In this section, I'm going to talk to you about aromatic compounds and what those are and what they mean. And some of the general properties for these types of compounds is that they are going to be cyclic and they're going to have double bonds in them. So one prime example of a aromatic compound is a benzene ring, and this has a formula of C6H6. And of course, when we write these out, we are not going to generally include the hydrogen at each of the corners here. So typically that's drawn without them, just because, again, it just makes it cleaner and easier to see. But we of course know that when we draw these skeletal structures out, that there's an inferred, we know that there are hydrogens at the corner, we just typically don't draw those out like I just did. So typically we're going to just see that as a ring like this. So there's just different ways of doing this. Kekule is basically telling us that we have every other carbon has a double bond. And you can see if I use this, kind of show you, the electrons will bounce to these other bonds. There is kind of a free flow of electrons, and then you can see that double bond is kind of, these double bonds move to these different positions within that benzene ring. So that's just kind of showing you the changes and how those move. The Robinson structure is the same thing. It means the same thing, but they just kind of draw a circle in the middle just to show you, or to kind of represent that movement of those electrons constantly and having that space all around that ring. Typically, when we're going to look at the chemical reactivity, there's really only one type of reaction that these undergo, and it's substitution. And I'll go over that quickly and briefly with you. It's not something that I'm too worried about in terms of this class covering. But we also know that it has sp2 hybridization, because those p orbitals are where the electrons are going to be utilizing on each of these carbons to flow on the top and the bottom. So they're actually flat structures. There's something else that's interesting about this with this resonance structure. I'm going to show you this kind of chart here. And this is represented on the y-axis is the amount of energy. So the potential energy of that system. So the higher the potential energy, the less stable it is, and the lower potential energy, the more stable it is. So we can see that the most stable structure is actually the hexane, which is just a six-membered ring without any double bonds. As soon as we add a double bond on here, we get a little bit more energy that's involved in that, because it restricts the structure a little bit. It restricts where those electrons are, and the shape isn't as free to kind of move around between the different chair positions. For example, these kind of have some flexibility in them. But that's a little bit lost when we add that double bond. If we add another double bond on an adjacent carbon, we get that increases the amount of potential energy in this system. And if we add a third one, this is the theoretical or the hypothetical amount of energy that this would be involved in if we didn't have resonance. But the resonance idea, and that's the idea where these electrons are flowing throughout this whole space. So we save some energy because we give electrons more area to move. The actual resonance energy is about 152 kilojoules per mole of this molecule, which brings that energy level kind of intermediate between a single and a double bond within this ring. So this is actually a much more stable compound than we might think it would be, because we've added that stability due to allowing the electrons to flow throughout this ring and provide that resonance ability. So this is a conjugated system where we have a double bond, a single bond, a double bond, and a single bond. And this conjugated resonance system is what reduces that energy level for these types of ring structures. We see these structures in a lot of biological compounds, and they're part of the capsaicins or capsinoids, which is the heat structure that gives us those Scoville heat units for different types of peppers, for example. Nutmeg, beetroot, that gives it the color. We have different chemicals with coffee and tea and chocolate, caffeine. All of these different things are going to have these different types of aromatic rings as part of them. And here are just kind of another list of different components of vanillic acid, caffeinic acid. A lot of different types of flavor compounds are also have these ring structures as part of it. And obviously there are other things attached to them, but this is kind of one of these interesting fundamental components of a lot of the flavor compounds in our foods. So I mentioned before that these are sp2 hybridized. Now, most carbons that are just have single bonds, if I have something like a ethyl group with a carbon and we're going to attach it to another carbon, when these are attached to four things, typically these are sp3 hybridized. Remember, they have four different sp3 hybrid orbitals that if we have a carbon, we would kind of draw sp3, and then we'd have another one sp3, and we'd have another one that's going to come kind of out into the plane of the computer screen, sp3, and then we have one kind of going back, which would be an sp3, and this would be a tetrahedral. So this is a typical carbon is going to be sp3 hybridized. We're going to have four different sp3 orbitals in those four different orientations. But when we create a double bond, these pi bonds, they are generated from p orbitals. So we have to subtract out one of those sp orbitals. Here we'll have one orbital that's going to be sp2. We'll have another orbital that is going to be sp2, and then we'll have a third orbital that is going to be sp2. So those sp2 orbitals are going to be the three new orbitals that are going to attach with these sigma bonds. So for example, you'll notice that this carbon here is attached to a hydrogen and two other carbons. So here, let's say we have another sp2 hybrid orbital carbon. This would overlap. That's our double bond. That's our single bond, our sigma bond. We'll have another sigma bond over here with carbon, for example. And then we also have hydrogen here, which has an s orbital. Here's another sigma bond. So that's the representation over there. Now we also have the unhybridized p orbital. So since we have s and two p's, there's still an extra p orbital that hasn't been hybridized. And these are going to be above and below that plane here. And that's what's represented in these blue orbitals here above and below the plane. Now, when we have two of these side by side, these two carbons here, you can see those unhybridized p orbitals here and here. Those are going to interact on the top and the bottom. And you can see that represented here. That's going to kind of allow those electrons to flow in that whole space. Now, because we have all those unhybridized p orbitals around this whole ring, we're able to kind of create this kind of donut above and below the ring where all of these electrons can flow around and move. And that's this idea of resonance, and that's the idea of an aromatic compound, because we have a lot more room for the electron to flow, and it stabilizes that whole structure by quite a bit. So these ring structures are not just a typical alkene, they're actually a lot more stable because of that aromaticity, that conjugated system of electrons where we have the double bond, single bond, double bond, and allows those electrons to flow above and below. So something to note about aromatic compounds is that they're cyclic, so they have a circular structure, they have a ring shape to them. They're planar because they're sp2 hybridized, so they're that trigonal planar shape and they're kind of flat. And that again allows those electrons to flow on top and below that plane where those other sigma bonds are. And they're conjugated, so that those electrons can move throughout that system. We're not going to get into this. There's a rule, there's Huckel's rule that define if it's aromatic or not. Not something we're going to worry about necessarily, but we're going to just look at a few examples here just to kind of get an idea if something would be aromatic or not. So here, which of these would you expect to have significant resonance energy? And so what we can do is we can look at that ability to have, okay, so here's a double bond, here's a double bond, we have a single bond in between. So we would have, these would be conjugated systems of pi electrons. Now these lone pair, interestingly, if it's beneficial to that molecule to have those in an unhybridized p orbital, it will happen and they will be in that space. So this nitrogen would actually unhybridize these electrons into the p orbital to allow it to be aromatic. So this one would actually have some resonance structure. It would be aromatic and it would work in that way. These electrons here, we have a double bond, and a double bond, all separated by single bonds. That would be aromatic. And then, of course, benzene ring, which is shown here. We've already talked about that. That is also aromatic. So these are all examples of aromatic compounds. So just to reiterate again, this is the idea where we have energy levels, and this difference between the theoretical or hypothetical amount of potential energy and what it actually is because of that resonance stability. And the analogy I like to use is if we have an orbital, electron orbitals can have typically 2 electrons in them at any given time. So when we talk about orbitals having 2 in them, usually it's kind of like a house or an apartment, right? So just because if you have a 4-bedroom apartment and you have 4 people, they'll typically like to split up into those 4 little different bedrooms. But if you only have a 2-bedroom and 4 people, then they're going to have to bunk up, right? So this is the idea, though. It's basically the idea that we're going to have a larger house. So you'll have more people in there, but you have more room, so that makes all of those people more happy overall. So they're able to share more of that space in your house, for example. So that lowers the energy, that makes the electrons happier. I know it's not a perfect analogy, but that's kind of how I think about it. So anytime you give electrons more space to roam in and still allow them to be close to the nuclei of different atoms, then they're happier in that state. So this is the difference in resonance because of that conjugated system compared to, for example, isolated electrons. So this actually has some effect in terms of their ability to interact with other elements. So if this electron set is going to be involved in a chemical reaction, you would actually break that resonance stability. So this a lot of times doesn't really like to happen. So we have to really make sure that we're going to typically, if we do a substitution, if we are going to remove that proton and add something else, we have to really make sure that the action is going to actually restore that resonance because it's going to be energetically favorable. So we typically aren't going to just lose that and then have only 2 double bonds. It's not going to work that way. So we're going to only do substitution reactions with these types of aromatic compounds. We're not going to spend too much time looking at the different types of substitutions. In terms of organic chemistry, when we name these, we call them electrophilic aromatic substitutions, or EARs, and those are going to, when we're basically substituting a proton, you can see that here is going to leave, and then we are able to reform that resonance structure. Just in terms of also just some vocabulary, when we're talking about where things are happening, based off of, for example, something that attached, that attachment point, if it's right next to that attachment point, that's called an ortho position. And that's, again, relative to that attachment of whatever we're talking about. If it's, we'll go through some examples of things that can be attached to benzene rings, but the ortho position is on the just adjacent to that. So that could be at this position, or it could be at the other position. There's para, which is on the other side of that attachment point. And then there is going to be also meta, which we actually don't have here at the moment on this slide, but this is the meta position. I always like to think of this as ortho. You think about an orthodontist is really close to your face when they're working, and these 2 positions are really close to that bonded electrophile. The para position is really far from the other, that bonded electrophile. So its para is far away, and then meta is in between the two. Here are some examples of attached electrophiles, or things that can be attached to a benzene ring. We have a OH group, and this whole thing is called a phenol group. If we have a methyl group, it's called toluene. Benzene is just where that standard hydrogen. Chlorobenzene is where we've attached a chlorine or substituted a chlorine at that position. And nitrobenzene is where we substituted a nitro group for that proton. So what we actually know is we can actually do subsequent substitutions. We can substitute things at other areas. And if you look at some of these other compounds, you'll notice obviously these have multiple substitutions on them to make these vanillic acid, for example, right? So this has to be possible. And so these things can be attached at different areas, and you can synthesize this chemical relatively easily in the lab. But of course, these synthesis reactions, these things are also happening in our bodies as well, or in plants, for example, and they're also able to be synthesized in those areas. But for organic chemists, what's interesting is when we're trying to synthesize or use these as starting points for adding other things, some of them work better in terms of how quickly that reaction will go. So if I take a phenol and I try to do a substitution reaction, it goes 1,000 times faster than a benzene. And if I take a nitrobenzene and try to react it with something, it is over 1,000,000 times slower than benzene. So your starting material is going to make a difference. And what is actually happening here is kind of interesting. If we look at a heat map of the ring after these substituent groups have been changed around, you'll maybe be able to see what's going on. So here are some examples. We have just the normal benzene ring, and this is a heat map, is telling us where these electrons are located. And here the benzene ring, it's around this ring where we have those double bonds. If we take a methyl benzene, we took a methyl group here, that ring is similar in terms of the kind of the heat that's coming out of it. If we look at aniline, where we have a nitrogen group, we can see that some of that structure is kind of the electron density is up in this nitrogen now. And nitrobenzene compared to benzene, this has gotten a lot more green. So these are the things that this is going to, when we pull electron density out from this ring, that slows the chemical reaction down a lot. And when we add, for example, the phenol, we would suggest that would add some density here and provide this more red part of this. And that's because these electrons are the things that, if we have a high density, they're going to want to move and interact with other things. So that's what's actually driving the speed of those chemical reactions with these aromatic rings. So where else do we see these things? Naphthalene, anthracene, phenanthracene, pyrene. There are a number of different polycyclic compounds where we have these multiple fused rings, and these all obey that rule that I mentioned with Huckel's rule and how many electrons. You don't have to worry about that. But some of these components can be environmental toxins. They can come from tar or smoke, in terms of petroleum smoke or burning those things. Even some smoked meats can have really low amounts of these. But we can see these things in various regions of the environment. Naphthalene, sometimes used as mothballs, for example, as a moth repellent. So they do have functions as well. We can also see there are some really interesting things that we can do with these in terms of putting these together into kind of carbon nanotubes, and for drug delivery and other kind of purposes and functions that are being explored in chemistry and in medicine as well, in terms of delivery for certain compounds. So you can see these are some interesting shapes that we can make with these things, and they're very stable, and they also have interesting properties for conductance and strength. Here are some interesting compounds that we'll talk about perhaps later in the semester when we get into the biochemistry, where we have a lot of different purines that make up DNA base pairs, different structures that are going to be found in biological compounds in terms of their ability to interact with different metals, and these are kind of foundational components of a lot of biomolecules. So here's an example of some of these aromatic rings that we were talking about in terms of treating cancer, for example. So this is you might notice this shape that we just saw a little while ago is similar to that pyrene molecule, and it's attached to a sugar, which is attached to this pyridine compound, which is also aromatic. These structures can be used to treat cancer because they're able to intercalate into the DNA for really quickly replicating cancer cells. So it actually helps to slow down that regenerative or that replication process for cancer cells. And this is one of those things to slow down that cancer growth by inhibiting that DNA polymerase activity. So these are really important structures in biology that utilize this aromatic shape. And that's all I really wanted to talk to you about in terms of this chapter, just to kind of give you an idea and an introduction into aromatic compounds. The stuff in terms of the synthesis and the how we synthesize these in organic chemistry, we're not going to really get into too much detail about that. That's just kind of just some other extra, hopefully interesting stuff for you. But really what I want you to know is that these things are common in biology, and they have some extra stability that allows us to kind of build off these things and create other molecules. So that's all I'm going to talk about in terms of this, and that concludes the chapter 13 content regarding double bonded carbon-based compounds.